So, what's all the buzz about Snodgrassella-Alvi? This presentation will look into the symbiotic relationship between the honeybee and its symbiont Snodgrassella-Alvi. With this specific system, there is reason to care for it as it is a model organism that's useful to study for gut microbial communities. And it does promote bee health, and bees are a major pollinator in the world, which as you can expect, indirectly helps us as well with agriculture production. The host in this relationship is the honeybee, and it comes from the phylum Arthropoda. The class is Insecta, the order is Hymenoptera, the family is Apidae, and the genus is Apis. So overall the species is Apis mellifera. It does need pathogenic defense via biofilms, which I will go into further detail later. And it also needs successful colonization of gut microbes in order to sustain an anaerobic environment within its gut. The star of the show is the symbiont Snodgrassella-Alvi, and it comes from the phylum Tsunomonodota. This specific symbiont needs a nutrient-rich environment, a stable habitat, and a means of transmission, which is horizontal, which I'll discuss later. And it needs all of these things as its loss of autonomy no longer allows itself to sustain itself. So this symbiont is transmitted through horizontal transmission, specifically through anal trophallaxis or anal grooming, basically through a fecal route or exposure to fecal matter. Now this isn't fully 100% confirmed, but it's the most likely answer that researchers have gathered. As during the pupa stage when honeybees emerge, they actually emerge without their core gut microbes or very few bacteria. So they're going to be highly dependent on healthy hive mates to acquire their symbiont. So when looking into horizontal transmission for the symbiotic relationship, I found this paper with this figure that I feel best explains it. Essentially, the main takeaway here was that anal trophallaxis or anal grooming was the main route of transmission. And to simplify this acronym, NEWS is basically newly emerged workers. So they had about a total of 5 groups here, each one containing at least 10 bees. And to essentially break down what they are is that we have newly emerged workers exposed to hive material, we have newly emerged workers exposed to nurses and hive material, we have newly emerged workers exposed to fecal matter from the hindgut, and then we have newly emerged workers exposed to just nurses. And the last group being newly emerged workers exposed to oral trophallaxis only. And the way that they achieved this was by putting 5 newly merged workers and 5 nursing bees, and they separated them with a barrier in between the container with a circular opening that was the only means of contact for the bees through their head. And so essentially what they saw here is that when they exposed newly emerged workers to either hive material or oral trophallaxis only, what we actually see here is the accumulation of non-core taxa from the gut microbe of the honeybee. I indicated in the red circles that that's what S. alvi is, this like yellow mustard color, and it's clearly absent or very little in either exposure to hive material or oral trophallaxis only. But then when they look at newly emerged workers exposed to, for example, nurses or fecal matter, you see a rise in S. alvi and the actual accumulation of core gut taxa for the honeybee. So the researchers here in this paper concluded that exposure to fecal matter was the most likely route of transmission, so either through trophallaxis or through grooming with healthy nestmates. So as far as occurrence is concerned, when it comes to the host, the honeybee, like most hosts for symbionts, they provide shelter and a means of transmission for the symbiont, as well as food, and in this case it's host-derived organic acids. And the SLMI will provide pathogenic defense via biofilm formation or strengthening of the immune response. And this is a pretty ancient symbiosis that has been estimated to be around for, or co-evolved for around 80 million years. Now, in terms of recognition, S. alvi essentially chooses the bee as it's very specific to just the honeybee and its gut, and specifically the ileum of the honeybee gut, as it can't really live outside of the honeybee for very long. And essentially what it uses type 4 pili to initiate attachment and begin biofilm formation. Now, the type 4 pili is specific to the honeybee, as I mentioned, though the exact recognition sites aren't really known or identified at the moment. Um, but yeah, so through this adhesion in a suitable environment, it can begin the biofilm formation. And as far as the environment that it's in, it needs to be microaerophilic. So it can live off of very low oxygen amounts, and it consumes it to create an anaerobic environment within the honeybee gut. And it also needs to live within an acidic pH. So with these conditions met, S. alvi can then thrive and then begin successful colonization. So another paper that I found that essentially helps support this finding is this paper right here, which had a really nice visual as well. And essentially, the main takeaway was that type 4 pili mutant genes results in reduced biofilm formation. So what they did here is that they created knockout genes for the biofilm formation, and it basically made them lack any sort of adhesion. And so without this adhesion, it couldn't successfully form biofilms on the ileum or the gut wall. And so without this biofilm, there was no means of any potential cross-feeding or feeding on host-derived organic acids. So it was unsuccessful colonization, and there was no recognition or attachment. So essentially, without the type 4 pili and their adhesion, no symbiotic benefit could occur whatsoever. And I essentially already alluded to this, but as far as establishment is concerned, its settlement involves dietary nutrients because, as I mentioned, the symbiont has no sense of autonomy whatsoever as it cannot live on its own anymore, so it cannot produce its own food. So it'll be essentially completely reliant on the host. It'll grow on host-derived organic acids like citrate or glycerate and things like that. And so this is what's key for colonization to even occur. So for influence on development, as I mentioned, S. alvi is essential for bee health and it promotes it in a very positive way. And this is done through colonization resistance via biofilm formation because it has this niche in where it takes up space in the ileum of the gut wall, in the ileum of the honeybee, and helps keep any opportunistic pathogens out. But it may also be involved in strengthening of the immune response as it can modulate tryptophan metabolism in the gut. So it's going to be responsible for breaking down kynurenine to anthranilate, which can be tied to immune regulation, as kynurenine is part of the pro-inflammatory response system, and anthranilate reduces inflammation. And by reducing inflammation in the gut, it can actually promote colonization of other core gut microbes. Or other bacteria within the gut that's beneficial to the honeybee. So this is going to be essentially S. alvi being responsible for rebalancing host immune metabolites. So these influences on development can actually be tied to even possibly larger developmental benefits. So kynurenine is actually going to be really important during the larval stage for the honeybee, but it can actually be associated with neuronal defects if it's overactivated or overstimulated. So if there's a surplus amount of it, it can actually lead to hyperactivity or motor defects in the honeybee. So S. alvi is going to be responsible for helping regulate and balance this out and preventing this from occurring. And anthranilate is actually a precursor to tryptophan, which can be responsible for things such as serotonin, tryptamine, or endoderivatives, which are part of anti-inflammatory responses. And there have been indications that this actually may even be linked to enhanced memory and learning within the honeybee, though this is relatively novel and still has to be looked into further. And also another benefit that S. alvi provides is that it can actually metabolize urea. So by doing this, it will actually remove a major waste product out of the honeybee system. Now, one of the cooler papers that I actually found was called Symbionts Shape Host Innate Immunity in Honeybees. And so essentially what the gist of this was that symbionts actually can affect honeybee immune gene expression, and they looked into live and heat-killed versions of S. alvi and how it can actually protect the honeybee from the opportunistic pathogen Serratia marcescens. And they can also help aid in expressing host antimicrobial peptides against Serratia marcescens. So within this paper, it actually had a figure that really helped explain one of the main takeaways. And essentially, the gist here is that live S. alvi have a higher survival rate. And so essentially what they did here is that they treated microbiota-free honeybees with either live S. alvi, heat-killed S. alvi, or sterile sugar syrup. And so on the right-hand side, we see that S. alvi typically did have a higher survival rate than heat-killed S. alvi, and all these findings here are statistically significant. But then when we look over to the differences between S. alvi and heat-killed S. alvi exposed to a pathogen, we actually see that without live S. alvi, they still did have a lower survival rate overall. And this mainly has to do with the fact that heat-killed S. alvi actually cannot colonize the ileum of the honeybee gut. And this is actually really problematic, as the biofilm formation is one of the main lines of defense that S. alvi provides for the honeybee. And so without this, it can't really aid in colonization resistance and keep opportunistic pathogens out. But when we compare heat-killed S. alvi plus pathogen compared to just exposure to pathogen only, we see that it still provided some form of protection compared to bees not exposed to any type of S. alvi. So they really wanted to look into what's going on with this as well. And so to help answer this, we look at the figure following this, and where the main takeaway was that live and heat-killed S. alvi help trigger upregulation of antimicrobial peptides. And so in this figure, we have red being live S. alvi, blue being heat-killed S. alvi, and green being heat-killed E. coli. And so what their findings helped tell them is that they're responsible for triggering host immune responses with AMPs. And the main reason is that the main reason why they even included E. coli to begin with is because they wanted to see whether this was symbiont specific or gram-negative bacteria. And based on the results, it's actually not symbiont specific at all. It's something that gram-negative bacteria within the honeybee gut are capable of doing, since E. coli had relatively similar amounts to antimicrobial peptide upregulation compared to the live or heat-killed versions of S. alvi. So even though it's not symbiont specific, this still does go to show that S. alvi is still part of immune responses against pathogens, and it's not just biofilm formations that they aid in. So as a means of looking further into this, they followed this with another experiment, and where the main takeaway was that when compared to live S. alvi, heat-killed S. alvi actually trigger a more extensive immune expression response, as I indicated with the red circles on the figure on the right-hand side. And it's possible that live S. alvi already contribute with biofilm formation, as this symbiotic benefit already helps suppress pathogenic infection. But moreover, it is also the reason that this is occurring could be due to the fact that heat-killed cells cannot colonize or form biofilms, so their molecular components like lipopolysaccharides and things like that are more freely exposed to recognition receptors throughout the gut. And in addition to this, because heat-killed bacteria aren't anchored or growing in a biofilm niche, their immunogenic molecules are more likely interacting more broadly with host immune sensors, such as Toll and LMD pathways across the gut tissue. Moreover, the live bacteria or live S. alvi may potentially be reducing the magnitude of LMD pathway gene upregulation that heat-killed cells would otherwise cause, implying that live symbionts might modulate or tone down host immune signaling compared to inert bacterial debris. So essentially what this is saying is that, or what I'm trying to say is that live S. alvi interacts with the host in a co-evolved way with its biofilm that helps essentially reduce immune activation, whereas heat-killed S. alvi is essentially just entirely unregulated in the sense that its compartments are completely exposed to the host immune system, which allows it to interact more directly with it, and which helps elicit this broad transcriptional immune response. So a second paper that I found regarding immunity in the honeybee and S. alvi is where they actually engineered symbionts to help activate honeybee immunity and limit pathogens. So here in this paper, what they basically did is that they took the wild type version of S. alvi, primarily found in the ileum of the honeybee gut, and engineered it with a plasmid. And so this allowed it to produce double-stranded RNA or dsRNA using two inverted promoters. And so they wanted to see whether this newly engineered SLI can recolonize the honeybee in the way a regular SLI could. And so essentially, yes, it was able to do so. And to keep this figure short and sweet, is that basically shows that yes, the engineered SLI was able to successfully colonize the ileum of the honeybee gut and remain functional. And I really like this figure down here at the bottom hand right that I circled in red, and where it shows that the engineered symbiont SLI was able to colonize the same localization that the wild type SLI was able to do. So they were successful in actually doing this, which I found pretty neat. So then once colonized, this was actually able to continuously produce double-stranded RNA within the bee. And by doing so, it spread beyond just the gut, which was responsible for upregulation and differential expression of immune pathway genes from the host to essentially activate RNA interference, right? And so this is actually really crucial because RNA interference is a natural cellular process that silences specific genes, and this actually helps act as a defensive mechanism against viruses by targeting and degrading viral messenger RNA to stop their protein production. So then in this paper, they were essentially wondering, can this be used to silence specific host genes and not just genes from invading pathogens or viruses? And so to answer this, they basically engineered SLI with a double-stranded RNA plasmid that was meant to target the insulin receptor of the honeybee. And so what they saw is that it did actually lead to reduced expression of the receptor. And so this resulted in the honeybees actually gaining more weight and thus increasing their feeding behavior. So this finding basically told them that symbiont mediated RNAi silences bee genes and impacts behavioral and physiological changes within the honeybee, as indicated by the weight gain and increased feeding behavior. So then, now knowing this, the follow-up to this was, can symbiont produce double-stranded RNA protect against viral pathogens and parasites? And the short answer is essentially yes, because when they designed a double-stranded RNA against the deformed wing virus, or DWV, and parasitic mite varroa, they found that those harboring the engineered SLI actually saw suppressed viral infection for deformed wing virus, and any mites feeding on the bees died at a faster rate. So they were clearly meant to target this viral pathogen and parasite, and were successful in doing that. So this was essentially a novel tool to help improve honeybee health. Essentially, the TLDR of this is that engineered SLI can induce eukaryotic RNAi immune responses. And I thought this was really cool just because this was like I mentioned, relatively novel and not something seen before. The implications of creating a new SLI symbiont to help target these problems is definitely food for thought, just because when creating new things and putting them out into the wild, we never really know what the implications of that may be or any consequences. So I just think it needs to be further looked at before it can actually be fully implemented in real-world scenarios. Dysbiosis of the honeybee system can actually be triggered by several things, and one of the main ones being pesticides, and the one I'll discuss is glyphosate, as it does actually impact it in many ways, as well as parasites like parasites such as Nosema ceranae. We have diet shifts such as age diet, which are typically found during seasonal changes when, such as winter, when food production is very scarce and the honeybees have to rely on a stored honey and bee bread, for example. As well as pathogens like Serratia marcescens and Frishella parara. Dysbiosis can actually influence the host fitness in a couple of ways, such as impaired development, increased mortality, susceptibility to pathogens, and unbalanced microbial communities. So then over here, I just basically implemented a graphic of what glyphosate looks like with its molecular components. But for impaired development, it can actually impair spatial processing of the honeybee. In an experiment that I'll get into further detail, when exposed to glyphosate at a high amount, some bees actually failed to return back to the colony, and they lost them essentially in the experiment, and that's how they kept track of that data. But moreover, it actually perturbs honeybee gut bacterial communities, which I'll get into the next slide. So then with this figure for impaired development, there were three groups here, and where the control group, the honeybees were exposed to sterile sucrose syrup. The second group, which is G5, they were exposed to 5 milligrams per liter of glyphosate, and just for context, glyphosate is found in Roundup. And also the third group being G10, where essentially they were exposed to 10 milligrams per liter of glyphosate. And to keep track of the bees, they were also marked with paint. And the glyphosate levels here were essentially meant to mimic environmental exposures that they would naturally encounter when foraging. And so the big takeaways here on this figure was that S. alvi was actually one of the core gut microbes that was significantly impacted in a negative way, and where we see here in green that between day 0 and day 3 of G5, it was significantly reduced overall. And then over here on absolute abundance, we see here that between day 0 and day 3 again, that the overall core gut microbes of S. alvi was significantly reduced. And this does include Snodgrassella alvi. After G10, they really didn't have an explanation as to why it remained relatively unchanged, but it just goes to show that 5 milligrams per liter glyphosate is enough to cause great disturbance for the symbiotic system of the honeybee. So with this figure, they essentially help explain that glyphosate does actually lower the abundance of S. alvi content throughout the honeybee gut. And so because it specifically targets S. alvi, this can actually really hurt early gut bacterial colonization. Because it interferes with normal colonization of S. alvi. And S. alvi is actually known to cross-feed into other microbes of the honeybee gut. And by doing so, it will help other core gut microbes successfully colonize and form their own biofilms. So essentially, it's a domino effect in where you lose the benefits of S. alvi, you lose the benefits of many other microbes within the honeybee gut. So then naturally, as one can assume, this will actually lead to increased mortality of the honeybee. So as established, glyphosate exposure at a certain amount will actually reduce the protective effects of S. alvi, almost making them non-existent to a point where opportunistic pathogens will actually be very happy about this, as they now had room to invade and infect, right? Because there's no longer this colonization resistance provided by S. alvi. So there's going to be the loss of colonization resistance through the loss of the biofilm, and thus the loss of cross-feeding, as I mentioned, because this is going to disable greater protection barriers provided by other core gut taxa. And then another paper found that exposure to Nosema surinae and other pesticides combined will actually lead to further gut dysbiosis and naturally decrease honeybee survival as well. So then continuing our talk about glyphosate, it actually does make the honeybee much more susceptible to pathogens, and in this case, Serratia marcescens, right? So then on the figure on the right-hand side, I've established that glyphosate exposure essentially leads to reduced S. alvi, and the reduction of S. alvi leads to reduced protection from pathogens such as Serratia marcescens. So on the right-hand side, the top layer is essentially the honeybees exposed to glyphosate, and exposure to glyphosate itself doesn't necessarily decrease their survival rate, but then the line in the middle was the honeybees exposed to Serratia marcescens. And so when exposed to Serratia marcescens, they can still survive for the most part, but there still is a decently high mortality rate. But then this is further exacerbated by the fact that they wanted to see if glyphosate exposure, in addition to Serratia marcescens exposure, led to increased mortality rates, in which it actually does. As bees exposed to glyphosate and Serratia marcescens had a very, very low survival percentage, right? So it just goes to show that glyphosate did have something to do with this, as it targets an enzyme called EPSPS within the within S. alvi. And specifically, S. alvi contains the sensitive class 1 version of EPSPS. Um, and so the reason that it's susceptible to glyphosate is that the glyphosate molecule can bind effectively to the active site of class 1 EPSPS and essentially block the enzyme's function, right? Um, and because it's so easily inhibited by glyphosate, the bacterium of S. alvi cannot produce any essential aromatic amino acids that it needs to grow and function. So naturally, this will lead to a reduction in the abundance of S. alvi in the honeybee gut. Um, and because there's a reduction, as I mentioned, this naturally leads to the lack of a protective biofilm, and that's how this dysbiosis occurs and leads to increased susceptibility to pathogens. So I also found another really interesting paper and where they observed Nosema surinae infection, and it actually led to a decrease in lactic acid bacteria. So naturally, with this spot being empty, niches have to be filled in by other core gut microbes. And in this case, S. alvi took on this role. And so what they saw was an increase in S. alvi, but they did note that a specific strain did decrease. And at first I was thinking that shouldn't this be a good thing, right? Because S. alvi is a beneficial symbiont of the honeybee, and so I thought more of it would be a good thing. But then I realized that it's good until it isn't, because this biosis is still going to occur. When it comes to microbial communities for the gut, for any organism for that matter, we need to prioritize diversity and abundance of multiple microbes and not just a singular one, right? But this biosis still occurs because there's there's an unevenness or an unbalance of microbes in the honeybee gut. But it's also important to note that not all strains provide the same function. So for all we know, this strain shift actually impacted the honeybee gut in more ways than one, because it may have actually lost a beneficial strain that provided the symbiotic benefits. So yes, while it may have gotten more S. alvi, it doesn't necessarily mean this is a good thing, because the S. alvi that it did gain may not have been the strain that it even needed to begin with. And then lastly for dysbiosis, another paper looked into age diets for the honeybee gut. And it kind of did multiple things at once. It caused dysbiosis and impaired development and increased mortality. So essentially what we see here on the right-hand side figure is that the dark columns were actual pollen, whereas the light columns were substitute pollen, right? And so they had either fresh pollen or fresh substitute pollen, or aged pollen or aged substitute pollen. And so what we see here is that when S. alvi decreases, it actually leads to an increase in the pathogen Freshella parara, right? So these are inversely proportional to each other. As one decreases, the other one increases, and vice versa. If there's an increase in S. alvi, there's a decrease in the pathogen, naturally through its defensive mechanisms. But it also leads to impaired development here. So we actually see that those who ate aged fresh pollen or or aged substitute pollen actually saw an increase in their mean thorax weight. So those eating an aged diet will uh, lack proper weight in that sense, right? And then for increased mortality, it's pretty significant in the top left-hand corner, and where those who ate a aged diet of pollen or substitute pollen saw an increase in mortality overall. And I think this relatively makes sense due to the fact that bees who eat aged diets, like for example those who have to eat stored honey or stored bee bread in the winter, they have to rely on other bacteria like Bartonella to help support their protein deficient diet. And because they're lacking the proper nutrients, it's going to leave them more susceptible to things like infection. And this is what helps cause this increase in mortality for the honeybee. So there are a couple ways of returning to equilibrium for the honeybee gut. And one of the main ways is going to be social transmission. And I've established this previously in where horizontal transmission is how it gains its symbiont of S. alvi through anal trophallaxis or anal grooming. And these are the most likely reasons. And this is due to the fact that they're surrounded by healthy nestmates, right? And they're going to essentially act as microbial reservoirs that will help reintroduce the proper strain of S. alvi into those who are lacking the strain. And this is further supported by another paper that I found, in where they actually did fecal transplants directly to from nursing bees to uh, honeybees that were lacking the S. alvi strain. And so what they found is that they actually were properly reintroduced to all core microbes, because when they compared the microbial gut communities, they were pretty much one-to-one. And so this helped reestablish this claim, right? There are also dietary supplements that can help uh, help the system return to equilibrium, such as phytochemicals, which are naturally found in nature in nectar, honey, and pollen. And so an intake of these through foraging can actually help the honeybee gut um, return to its proper state, right? And so when experimentally given this as well in a lab, they essentially saw an increase overall of microbial diversity, and which was very beneficial to the honeybee. So, funnily enough, I do realize I forgot to put words into this slide, but nevertheless, in short, the honeybee-Snodgrassella-Alvi symbiotic relationship has really shown us a lot in terms of what it looks like for a ancient, highly conserved model organism for gut microbial communities, right? Because through S. alvi, the honeybee and its gut, its health are promoted, and is able to successfully thrive and live on. And through the success of the honeybee, it's able to contribute to the Earth's pollination and agricultural production. And in terms, it really benefits us, right? It makes us think that symbionts are so little, but they provide so much value to our everyday lives, right? And it's not just for the honeybee Snodgrassella-Alvi. There are a multitude of symbiotic relationships out there that are beneficial in their own ways. But for this system in particular, we see that S. alvi provides not only defense via not only pathogenic defense via biofilm formation, but through means of removing things like waste products like urea, or aiding in upregulation and antimicrobial peptide production. And there are even studies investigating how to make S. alvi potentially even better through man-made engineered versions of S. alvi, which I think are really, really interesting to look into, regardless of what implications they may have. But yeah, so overall, the honeybee Snodgrassella-Alvi is a really interesting symbiotic relationship that needs further research, as it is all relatively novel within the last 15 years. And I think through further research, um, uh, it can establish itself as a well-defined gut microbial system for future symbiotic gut relationship studies. And then here are a list of sources I gathered, and links to them if anybody is interested.